Hey all, this is MedBite here, the channel where we break down complex medical topics into bite-sized pieces. So today I'd like to talk to you about bacteria, the structure and its classification. This is going to be a two-part lecture. So this video is going to be part 1, part 2 will be coming out tomorrow. Here is the simplified structure of a bacterium. A bacterial cell contains a capsule, which is a slimy protective layer surrounding the bacterial cell, and it's also located outside of the cell wall, so be aware of its location. The capsule is made up of polysaccharides, or sometimes proteins. Now, an important point that I'd just like to add here is that the capsule is not present in all bacterium, so be aware of that. Next, we have the cell wall. In bacteria, the cell wall is made up of a substance known as peptidoglycan, which is also known as murin. The cell wall has a simple role. It surrounds the plasma membrane and helps maintain the bacterium's shape and integrity. Now, also in bacteria, it's important to be aware that the majority of genomic material is contained in a circular molecule of double-stranded DNA known as a chromosomal DNA. On this diagram on the top left, you can see that the chromosomal DNA is a massive tangled mass. It's not a simple circle right here. You can see here that it's just a massive tangled mass of genomic material, and that's how the chromosomal DNA typically looks. Now there's another important distinction that I'd like to draw over here, and that is between chromosomal DNA and the nucleoid. The chromosomal DNA exists in the bacterium without being encased in nucleus. The chromosomal DNA is found freely floating in the cytoplasm, and so this term right here, the nucleoid, that refers to the region of the cytoplasm occupied by this chromosomal DNA. So be careful. Nucleoid does not refer to the chromosomal DNA itself. Many bacteria also contain extrachromosomal DNA. So this refers to DNA that is not part of the main chromosomal DNA. And this extrachromosomal DNA is often referred to as a plasmid. Plasmids are not essential to the survival of a bacteria, but they can carry additional genetic information that can confer advantages to the host bacterium under certain conditions. For example, plasmids often contain antibiotic resistance genes, which certainly do confer a lot of advantage to the host bacterium. Another very important thing that I will discuss a little bit more of later on is that plasmids can also be exchanged between bacteria. There is horizontal transfer of plasmids, essentially. Another thing I'd like to point out is that bacteria also contain ribosomes. So ribosomes are the site of protein synthesis, and in prokaryotes, these ribosomes are designated as 70S. 70S ribosomes. So 70S basically refers to the size of the ribosomes. In prokaryotes, you have 70S ribosomes. In eukaryotes, you have 80S ribosomes. Some bacteria also contain a flagellum, which is this faded shape right here. So the flagellum is a tail, kind of, and it just helps propel the bacterium. It enhances its motility, which is a fancy term for mobility. Bacteria may also contain pili, which are these hair-like structures located around the bacterial cell. They're kind of faded in this diagram. So there are two types of pili commonly found in bacteria. So the two common types of pili found in bacterium are fimbriae, also known as type 1 pili, and sex pili, which are also known as conjugative pili. So let's first talk about fimbriae, or type 1 pili. So fimbriae are short and numerous. They are involved in the attachment of bacteria to surfaces. For example, they can enable bacteria to adhere, which means stick to, so they can enable bacteria to adhere to host tissues, for example, the lining of the urinary tract or the intestinal epithelium. And by enabling bacteria to adhere to host tissues, bacteria can thus establish infection. So fimbriae are therefore very important in enabling bacteria to cause virulence, to cause infection. Now moving on to sex pili, also known as conjugative pili. Conjugative pili are longer but less numerous than their type 1 pili counterparts. Conjugative pili facilitate physical contact between adjacent bacterium, enabling bacterial conjugation to occur. Now, what is bacterial conjugation? Simply put, it's the process by which plasmids, which I mentioned earlier, are transferred between bacterial cells. Bacterial conjugation is a common mode of transfer of antibiotic-resistant genes contained within the plasmids between bacterium. So it is definitely a growing cause of concern with regards to the antibiotic resistance epidemic. How do bacteria replicate? Bacteria replicate via a process known as binary fission. So here I have drawn out a step-by-step illustration of this process, and I will of course explain. So we start off with our typical bacterial cell. I have omitted some of the parts which aren't relevant to binary fission, for example the ribosome. So here are the bare bone bits. You have the cell wall in grey highlight, you have the cell membrane, and you have one copy of chromosomal DNA. The first step in binary fission is DNA replication. So the chromosomal DNA is replicated first. And you can see here that we now have 2 copies of the original chromosomal DNA. In step 3, segregation of DNA occurs. Segregation is basically a fancy term for saying that the 2 copies of the chromosomal DNA move to the opposite poles of the cell. The word poles basically refers to a side. So this is one pole of the cell right here. This is another pole of the cell. So you can see that both copies of the chromosomal DNA are moving to opposite poles of the cell. Then in step 4, here is where things really start to happen. So over here, septum formation begins. So the septum is an invagination of the cytoplasmic membrane and the cell wall. So you can see in this diagram here, I have labeled the invagination. You can see that the bacterium cell is folding in on itself. In step 3, it was nice and square, but in step 4, the sides of the cell membrane and the cell wall are now moving into each other. They're about to touch each other. This region here is known as the invagination, labeled with a red arrow. So just to explain how invagination actually occurs, in bacterium there are a ring of proteins called the Z ring. So I've labeled that in blue right here, in this picture right here. So you can see that initially the Z ring is quite open, the diameter is quite large, the radius is quite large, but what happens is that as the septum begins to form, as the invagination begins to become more and more visible, the Z ring becomes smaller in size. It constricts, much like a snake. It constricts, and since it's attached to the cell membrane and the cell wall, it brings the cell wall and the cell membrane closer together. So that is the reason, the Z ring is the reason why invagination occurs. And eventually when the cell membrane and the cell wall touch each other, you get 2 copies or 2 identical daughter cells, each with their own copy, their own identical copy of chromosomal DNA as shown in step 5. So this is how binary fission occurs, and it's a very efficient process. It allows bacteria to rapidly reproduce and multiply under favorable conditions. Alright, so what are the different stages of bacterial growth? We've talked about how bacteria divide, but what are the stages of their life cycle? Well, let's say that we have a bacterial colony. This bacterial colony, or the growth of this colony, can be divided into several distinct stages, each characterized by specific changes in population size. Now, here is a diagram, an illustration of these various phases. Let me just explain the axis labels very quickly. On the x-axis, you have time, the time in which the colony is surviving for, essentially. And on the y-axis, we have CFU. CFU, as written in the top right-hand corner, stands for a colony forming unit. CFU represents the number of viable bacteria. So the higher the CFU, the more viable bacteria there are, the more bacteria that are able to survive and reproduce. So let's first talk about the lag phase. You can see here on the graph that the lag phase is characterized by rather a low population size, a low CFU. Now in the lag phase, the bacteria are adjusting and adapting to a new environment. In this phase, the bacteria are trying to increase their metabolic activity as they synthesize the necessary proteins to prepare for exponential growth. The population size remains relatively constant or may increase slowly. Next, you have the logarithmic phase, also known as the log phase or the exponential phase. In this phase, the bacteria experience a period of rapid and exponential growth. So in this phase, the conditions are quite favorable. The bacteria have synthesized the proteins needed for growth, and the population size as a consequence increases exponentially as bacteria quickly divide and replicate their DNA via binary fission. This phase is characterized by a very high rate of cell division and a low rate of cell death. Then eventually a stationary phase is reached. So you can see in the stationary phase, the population size is quite high, but it's no longer increasing as it was in the logarithmic phase. So in the stationary phase, the growth rate of the bacteria slows down and the population stabilizes. The rate of cell death is equal to the rate of cell division. Very important here. And then finally, you have the death phase. So because there are so many bacteria, all the nutrients have run out and there is a large accumulation of toxic waste products. And this contributes to the death of more bacterium. And as a result, the rate of cell death is greater than the rate of cell division. so the number of viable bacteria decline, and hence the colony forming units, or the number of CFUs, also start to decline. All bacterial colonies undergo a very similar life cycle. And of course, in various industrial processes, the aim is to try and maximize the length of the logarithmic phase and the stationary phase, whilst minimizing the length of the lag phase and trying to delay the onset of the death phase. Now let's talk a little bit about the classification of bacteria. Bacteria can be classified based on various properties. One important mode of distinction between different bacteria is morphology. Morphology refers to the study and characterization of the physical shape and structure of organisms, including bacteria. So in this example, bacteria may be classified based on their shape and arrangement. Common bacterial shapes include cocci, so if a bacterial cell is spherical, then that can be classed as a cocci, bacilli for rod shapes, spiral for helical shapes, and there are more. And common arrangement patterns include diplo for pairs. So let's say that bacteria or bacterial cells arrange themselves in pairs, then we'll call them or classify them as diplo. Strepto for chain-like arrangements, and staphylo for grape-like clusters. Let's give an example. There is a species of bacteria called streptococci. Cocci means that the bacterial cells have a spherical shape, and strepto means that these spherical bacterial cells are arranged in a chain-like formation. So you can use this principle in this example to extrapolate and classify other bacteria according to their morphology. Another very important mode of distinguishing between and classifying bacteria are staining techniques. The result that a bacterium produces in a staining technique can go a long way to help classifying it. Two very important staining techniques with regards to bacteria are Gram staining and Gill-Nielsen staining. In Gram staining, gram-positive and gram-negative bacteria are differentiated between. For example, the genus of bacteria Streptococcus is a gram-positive bacteria, whereas the genus of bacteria Escherichiae is gram-negative. On to Gill-Nielsen staining, this differentiates between acid-fast and non-acid-fast bacteria. I will be talking about these staining techniques in more detail later on, so for now just appreciate the fundamental principle that staining is an important basis of classification of bacteria. Another way of classifying bacteria is based off of their metabolic characteristics. So bacteria can be classified based on what they use as an energy source for their metabolism. In this case, you have four distinct categories: aerobic bacteria, anaerobic bacteria, facultative anaerobic bacteria, and microaerophilic bacteria, also known as microaerophiles. So aerobic bacteria require oxygen for metabolism, whereas anaerobic bacteria require a non-oxygen compound for metabolism. Now, just to be clear, anaerobic bacteria would not use oxygen even if it was present, because they require a non-oxygen compound to survive. Facultative anaerobes are kind of in between. They can become aerobic or they can become anaerobic depending on their environment and depending on the stage in its life cycle. So they can switch between both of their forms, their aerobic and anaerobic forms. Microaerophiles are a bit strange. They are aerobic, but they require a low oxygen level to survive. If they were to be placed in an environment where there is a very high concentration of oxygen, then they will die. They will experience enzyme inactivation, oxidative stress, growth inhibition, and they will die. So be aware of these metabolic characteristic-based classification for bacteria. Just to keep something in mind, we don't often refer to these forms of classification, or at least I won't be in this video, but it's important to be aware of this. Gram staining, as I mentioned earlier, is very important in distinguishing between gram-positive and gram-negative bacteria. Now, what is the difference, the structural difference between either types of bacteria? Well, gram-positive bacteria contain a plasma membrane, and overlying this plasma membrane, there is a very thick layer of peptidoglycan, which makes up the cell wall. So just imagine here that this is the cytoplasm of the bacterium. So you have a plasma membrane, and then overlying the plasma membrane on the outermost surface, there is a very thick layer of peptidoglycan. But in gram-negative bacteria, sorry for the incorrect spelling. So in gram-negative bacteria, you have a plasma membrane. Overlying this plasma membrane, you have a thin peptidoglycan layer. You can see how much thinner it is compared to the peptidoglycan in the gram-positive bacteria. And overlying the peptidoglycan in the gram-negative bacteria, you have an outer membrane, which consists of an important molecule, which I will be talking about more in detail later on, called lipopolysaccharides. So the lipopolysaccharide is an important antigen. An antigen is something that is capable of triggering an immune response. So the lipopolysaccharide, abbreviated to LPS, is an important constituent of the outer membrane of gram-negative bacteria, where it plays important antigenic properties, and it also enables the gram-negative bacteria to cause a wide variety of infection and disease. So now that we've discussed the structural differences between gram-positive and gram-negative bacteria, we can now better understand how gram staining works and why it produces the results that it produces. So in gram staining, the first step is to transfer the colony of bacteria that you're testing for to a microscope slide, right here. Once you've transferred the colony to a microscope slide, you then add a few drops of crystal violet stain. This is the principal stain. Now, what I mean by the principal stain is simply the first stain or the first dye that is added in a staining procedure. That's why the crystal violet stain is the principal stain. If it was another dye that was added here, then that dye would be the principal stain. Once you've added your crystal violet stain, you then add a few drops of Gram's iodine. Don't worry too much about the composition of Gram's iodine, just note the effect it has. Just note the effect that it has. You see, once you add Gram's iodine, you lower the solubility of the crystal violet stain. And by lowering the solubility of the crystal violet stain, you cause this stain to more strongly adhere, which means stick to the cell wall. So let me just quickly write that here. Adhere means stick to. Now, here is where I want you to recall the difference between a gram-positive and gram-negative bacteria. You remember that gram-positive bacteria have a very thick cell wall. They have a very thick layer of peptidoglycan, whereas gram-negative bacteria have a very thin layer of peptidoglycan. They have a very thin cell wall. So what happens next is that ethanol is then added to wash away crystal violet, which is not strongly adhering to a cell wall. So in gram-negative bacteria, since the cell wall is so thin, it is unable to retain as much crystal violet, and this crystal violet is easily washed away by the ethanol. But in gram-positive bacteria, where the peptidoglycan layer is very thick, it is able to much strongly hold on to the crystal violet and resist it being washed away by the excess ethanol. So this is why after you wash the bacterial colony with ethanol, you will notice that gram-positive cells continue to appear violet, whereas gram-negative bacteria are decolorized because they have lost the crystal violet stain because of their thin cell wall. And then finally, you add a counter stain, which in this case is safranin. So a counter stain, as I mentioned here, is a stain with sharp color contrast with the principal stain. Safranin is then added and causes the gram-negative bacteria to appear pink-red. And this is the mode of distinguishing between gram-positive and gram-negative bacteria. So that's it for today. Part 2 will be coming out tomorrow, where we actually start to classify bacteria based on their staining techniques, their metabolic characteristics, and their morphological features. So stay tuned, and if you enjoyed the content, then please do give it a like and consider subscribing. Thank you very much.